Laukik, check if Sridhar sir has joined. I think Shraddha has joined. Good evening, Shraddha. Thank you for joining.
Hello, hi, good evening. Thank you for having me in this session.
Thank you. We are waiting for Sridhar sir to join. Laukik, can you check with Sridhar sir once?
Yes, sir.
Just
I'm calling, sir.
quick question. I saw that there are other speakers too and wanted to know the chronology of the speakers.
Yeah. First, we have a chief guest who have already joined. So we'll have
Okay.
first chief guest address. Then you will be having the session. Then we'll have one more session. After your session, there will be one more session. So we have Rajavan sir, who is a chief guest.
How about Milind is here? Milind is also joined?
Milan is in other meeting, sir.
Ah,
But if he gets time, he will join. In the
okay.
meantime, he will be able to join because he is working towards that IPC.
I got you. I got your point.
Yeah, so he's busy in one meeting. So he said, we'll continue. We'll start
Sure.
with you. And then if he comes in, then we'll hold it for 5-10 minutes and then we'll have him
Great.
as his address.
Yeah.
And we'll
Fine.
go.
Fine with me. No problem.
So we are just, yeah, Sridhar sir, if joins, then I think we'll.
Yeah. Okay. Yeah. Let him start with whatever the round of introduction, and then I think we can go.
Correct.
So I'll wait for some more time. No problem.
Yeah, another 1 minute we'll wait, sir. Another 1 minute we'll wait.
No issue. No issue.
If Sridhar sir joins, we'll start in another 1 minute.
Hmm.
Yeah, Shraddha, we have received your PPT, so we are good to start. We'll be sharing the screen, so fine. Okay, so Laukik and Uma, we can start, I think.
Can we wait for 1 more minute, sir?
Yeah, no issue, no issue. Yeah,
Okay,
we are
1 more
waiting.
minute we'll wait because
Thank you, sir.
Rajavan sir has joined. So we'll wait for 1 more minute.
Let's Sridhar also join. We'll wait.
Yeah. So
Can you hear me now? I was not able to unmute myself.
yes.
Okay, good to know. And the camera also works now.
Oh, okay. Perfect.
Okay, sure.
Yeah, your background with the California bridge.
I'm a background of IPA.
Yeah, you are. I think Bob Shraddha ma'am has having a background.
No, I don't have any other option because
No issue.
usually we use Teams and the Zoom account I just created now, so no
Yeah.
other option.
No issue, ma'am. I think the pleasure. I mean, pleasure to talk to you, meet to you on online.
Yeah.
Same, likewise.
Thank
Yeah.
you.
Okay, I think Laukik will start. We'll continue with the introduction of speakers and then we'll continue looking into it. So welcome everyone for day 6 of a PV Leader Connect 2025 Global Pharmacovigilance webinar series. It's a 7-day webinar series to know the gateway of global journey in drug safety innovations, and also it's presented by Indian Pharmaceutical Association.
Mujib sir, Sridhar sir is joining in.
That's fine.
Yeah, we'll continue.
Yes,
So
sir. You can continue.
yeah, a gateway to global pharmacovigilance journey during the drug safety innovations. It's presented by Indian Pharmaceutical Association in academic collaboration with Clinosol Research Private Limited. So the meeting agenda is very clear. I'm going to invite today's speakers, and I'll be taking about what is PV leaders and all those connect series and all those things I'll be speaking. So what is an objective? Why we came up with the 7 days of intensive global pharmacovigilance series to enhance pharmacovigilance understanding. The series improves the knowledge of awareness about global pharmacovigilance practices and their importance in drug safety. That's one of the most important point. And we wanted to promote the global collaborations across all the world. We had multiple speakers from US and from different countries. It fosters the cooperations between among the different regions and also shares insight and strategies effective pharmacovigilance. And it addresses the emerging safety challenges. Most importantly, the pharmacovigilance comes up with safety challenges, the emerging safety challenges, how we can address it in a better way. We wanted to do it in 7 days of the webinar series. The structure, the 7 day duration, it was planned for whole 7 days, including Sundays. And daily we had thematic sessions. One day we had ICSR, one day we had Indian pharmacovigilance system, one day we had signals, one day we had AI kind of things. And it was having engagements and interactions. We took a lot of questions from YouTube. It's live in YouTube, and afterwards also we can watch this YouTube live. Only speakers are joining in Zoom calls, and it's live in YouTube so that everyone is able to listen to the sessions. And the theme and expected outcomes, the most important theme was regulatory updates, stay informed about the latest regulations. The second one is about signal detections and risk management. The third one was technology advancements and innovations, new enhanced technologies, including data analytics and automation tools. What was the expected outcomes to achieve the enhanced knowledge? Because most important is pharmacovigilance plays a lot of knowledge and global collaborations and regulations and also clinical knowledge. Build professional networkings and develop the actionable strategies for your pharmacovigilance. And the program creators, one, I think already Sridhar sir has joined, Dr. Sridhar from IPA Industry Divisions, who comes up with a lot of experience in pharmacovigilance. And the second person, it's me, CS Mujeevuddin. I'm the founder and CEO for Clinosol Research. And about the IPA, I will not take much time. The history of IPA founding purpose was for advances in pharmaceutical sciences and improves the healthcare outcomes. Committed for educations, focuses on research and also practices. It supports a lot of innovative research and best practices for enhancing the healthcare delivery systems. And major achievements and contributions of IPA. The education initiatives, the IPA promotes the pharmacy education to enhance the skills, knowledge, and also pharmacists nationally. And also the policy advocacy, they directly speak with the policies, improving the pharmacy practices in healthcare outcomes, and also they directly work with the government agencies. Research collaboration, IPA fosters the research partnership to advance the pharmaceutical sciences and practices. And mission, it fosters the pharmaceutical research and also industry research, promotes manufacturing best practices, and also supports the professional development. So that was the mission and objective of the Indian Pharmaceutical Associations, that how Indian Pharmaceutical Association has fostered the industry achievements. The key programs of industry collaborations, workshops, conferences, and also industry partnerships. The IPA has conducted enhanced quality standards, improved regulatory compliance, and also adoption of new technologies. And the one more hosting organization is Clinosol Research, leading edutech organization, building the skilled resources for clinical research and pharmacovigilance, led by industry expert to bridge the gap between academia and industries. Training, career counseling, corporate collaboration, academic collaboration, and industry connects. And this is a very brief recap of chief guests from day 1 to day 5. Day 1, we had Dr. R.N. Gupta, National President, IPA. Day 1, we had Dr. T.V. Narana, President, Sayer from Farm, past President, IPA. Day 2, we had Dr. Ramesh Adupu, Vice President, IPA, and also Vice President, FIP. And day 3, we had Dr. Vidyadhar, who is a professor and principal of CHIPS. And day 4, we had Subhash Mandal, National Honorable General Secretary of Mumbai, and also the Honorable General Secretary for Indian Pharmaceutical Associations. And we had Dr. Raj Vaidya, who is one of the most renowned person for community pharmacist. And then we had Mrs. Manjri Garath yesterday, in charge principal for KM Kundanini Pharmacy Polytechnic College, and past president of IPA, Indian Pharmaceutical Associations, and also FIP. And this is about the 5 days events, the chief guests and the guest speakers for day 1 to day 5. It was first one, Rajendra Kumar, Vice President of Global Head. Day 1, Sri Divya, CEO and founder of SafeVeg, day 1. Day 2, we had Deepa Arora, CEO of Clinosol, and also Priyanka Das, Associate Director of Global Safety. And then day 3, we had Call, Senior Director from US Amnil. And day 3, we had CS Mujibuddin, that's me one more time, I took one session. And day 4, we had Ranjit, CEO of QBD. And day 4, we had Sriram, who was from Clinical Development and Medical Affairs of GCPC vaccine. Day 5, we had Anuja, Country Pharmacovigilance Head of Kenyu. And also day 5, we had Rohini Pandey. She was the director for Abbott Laboratories. And coming back to today's topic, so the most interesting, we have 2 speakers today. Already Shraddha has joined, and we will be expecting Kakade sir to join shortly. So we are expecting one more chief guest. So I'll park it here. But we have one more chief guest for today. So a well-known personality, no introduction is needed. And very much everyone knows him, a motivational person and a person who has provided a lot of inputs for nationally and internationally. So let's welcome K. Rajabanu sir, who is a MPharm Director General of Pharmaceutical Export Promotion Council of India, which is also called as the PharmExcel. The PharmExcel is one of the organizations which helps almost all pharma companies. K. Rajabanu sir is a seasoned pharmaceutical and regulatory professional with over 3 decades of service in drug control and also vigilance, public health governance. A B.Pharm and M.Pharm graduate from AU, Visakhapatnam, he has begun his career as drug inspector in 93 and served across multiple districts overseeing licensing and also compliance and enforcement under Drugs and Cosmetic Act and drug prices control orders related status. His contribution in detecting violations and also launching prosecutions and also safeguarding the public health, establishing him as a key figure in strengthening the state drug regulatory system. He rose to senior roles, including assistant directors, vigilance, and assistant director, manufacturing, and also deputy director, where he led several landmark initiatives such as drafting good distribution practices guidance, CDSCO, implementing online licensing system, and geotagging medical shops, and also manufacturing units and blood banks, and creating the comprehensive drug monitoring systems, which is CDMS. As a nodal officer during COVID-19 pandemic, he successfully monitored or he maintained the essential drugs and also oxygen supply chains where everyone was searching for drugs and supply chains for oxygen. His expertise in further strengthened by WHO GMP, advanced vaccine trainings and participation in regulatory workshop organized by CDSCO, WHO, US FDA, UK MHRA, PDA, and global benchmarking initiatives. Appointed as Executive Director of PharmExcel in 2023, elevated to a Director General in July 2024, Rajabanu continues to champion the Indian pharmaceutical exports and global regulatory collaborations. He has sought after a speaker for training programs for drug inspectors, public prosecutors, and also active member in IPA, IPGA, ADCOG since 1995. His outstanding services has earned him the Best Drug Control Award in 2016, a Director General Best Performer Award in 2015, 16, 18, and 19, Certificate of Appreciation in 14, 15, representing India globally. He has visited different nations, including Ethiopia, Kenya, Nigeria, South Korea, and Uzbekistan, and also Vietnam, Italy, UAE, Turkey, Philippines, Malaysia, and USA on professional delegation, further international collaboration in the pharmaceutical sector. So without any delay, let's welcome Rajabanu sir to listen, and I'll hand it over to you, sir. Thank you for joining, sir. Thank you, sir.
Thank you so much. Thank you so much, Mr. Mujibuddin. I think for your kind words about me. Namaskar. Thank you so much. Very good evening to all of you. It's a great honor. It's a privilege to join you all today for this Global Pharmacovigilance Week celebrations, which you have already enumerated for the past 5-6 days, which has been 5 days rather. This is the 6th day. And in fact, I'm more privileged to be as a part of this program and also joining along with my good friend Professor Milanji Umaidkar and also Sridhar. And of course, I think recently just now got introduced to Shraddha ji also, and maybe the other speakers who will be speaking after me. I think more than what qualifies me to stand before you or sit before you talking is, I think, more than my qualification working in the drug administration and also in the from the regulatory also to the facilitator role. Probably, I think the last sentence which you have mentioned that I am also a IPA member. Yes, I am since 1985 as a student member, and I think from 1990 I am a life member of IPA also. So probably this rightly qualifies me to talk before you, all of you, address this very special occasion and the topic which you have chosen for the pharmacovigilance glow. And maybe the next part of it, which was also qualifies me to sit before you and talk before you, is in the capacity of a Director General of Pharmaceutical Export Promotion Council of India, set up by the Ministry of Commerce and Industry, Government of India, as our mandate to ensure that the promotion of all the pharmaceutical exports globally, which we rightfully earn the name Pharmacy of the World. In that sense, I think when we are talking about such a sector, pharmaceutical sector, which is a very national importance and very thrust area in the national importance for this one, and we all know that a drug is a very important, a very vital component in the healthcare system, and specifically at it affects the patient's safety also, and while it was treating also. So I think this occasion is not just a formal event, let me tell you. I think it is a reminder of our shared responsibility to safeguard every patient everywhere in the world. Now that because we brought the world and all, and we are supplying almost like our sector is almost like 60 billion US dollars, and half of that one we export to almost like 150 countries across the world. In that sense, I think probably over the period of years, 6 decades and all, the growth of the pharmaceutical sector and the exports have grown at a rate of CAGR 11% or something, which is very heartening to say. And when we export these to different markets and all, I think we also have this very important responsibility to make them understand that if there is something which is being hint that about the safety of the product, certainly we have to take note of that one. I think that's where I think the pharmacovigilance, what you call it, the global pharmacovigilance, what you recall, is more than a regulatory requirement, and it is a core and heartbeat of the patient safety and the bridge between innovation and trust, and the assurance that medicines do not just heal, but they do not harm also. I think the very cardinal principle of that one, that's why I think probably in the clinical trials and all, we all try to ensure that I think the benefit outweighs the risk and all. And at the same time, when it goes to the market, I think in a phase 4 and all, post-marketing surveillance and all, probably we have to understand that when we had in the clinical trial phases, probably we'll be looking at a very small group or maybe a very small group of the patients or even the healthy volunteers and all. But when it is being introduced into the market, when it introduced to the masses and all, then naturally probably the behavior and all has to be checked it. I think that is where probably the research organizations like yours, Clinosol Research Private Limited, are effectively working on those lines in the post-marketing surveillance and try to catch that signal in that sense and all through the professionals which are there, the prescribers are there and the patients are there. So in that situation, that's this year's the PV week that reminds us the safety is not just limited to our own domestic market, but is also the geographically to the entire world, where we are exporting almost like our 30% to the United States and almost like 50% both at the US and as well as Europe, which are very highly regulated markets, and also to the different parts of the Africa and Latin America and Asia and other things. So our mission finally seems to be the protecting the patients. So over the past decades, pharmacovigilance has evolved from being reactive to becoming more proactive and predictive. I think I come from a background of regulatory, and I don't want to take surprises. even when we are going for enforcement and all, we don't want to be surprised by an element in the market, whether it is a spurious, not of standard quality. So we try to upfront look at those kind of signals coming in the market. So is the case with this one also. So with advanced signal detections, artificial intelligence, I think a lot of digital tools, I think we as a country, we are known for IT, BT, PT kind of a things, information technology, digital technology, pharmaceutical technology, and biotechnology kind of a things. I think IT is one core things which I think we will be looking forward, and the digital health tools. And we are able to monitor, we can able to monitor risks in the real time and prevent harm before it actually occurs. So this is what we call, we are EHR. I think probably the country is gearing up for that electronic health records and all, and the kind of medications which have been already used and all, and maybe all that Ayushman Bharat and all those things and all. Some of those schemes which are there in the country, and otherwise also in the private sector also, the EHRs for the patients, and these are very important, and it captures. We can't work in silos. In that sense, I think probably a prescriber is somewhere and there is a dispensing and there is a patient taking them, and how much time it has been taken, what drugs have been taken, and these are all getting recorded and all. And it could be because of any of those reasons. It could be an idiosyncratic reaction, or it could be any of because of the drug itself, and which would not be detected during the clinical trials phases and all. And only at the PMS level we could able to find something odd, or some specific thing and all. You know that I think there could be a fixed dose, fixed drug reactions, fixed drug allergies and all, and it could be to a limited to a patient, or it could be masses, or it could be it can go to the fatal level also. We all know in some of those cases and also. So in that sense, we have to, as a collaboration is very important in that sense. Safety cannot be managed, as I said, that in silos. It requires a seamless partnership of clinical affairs, clinicians, regulatory affairs, what we call from this national regulatory agencies, as well as the state one, very medical, and quality of above all the vigilance of every professional who handles this or who touches the patient data. I think the very critical aspect of is that one. So I think even in India, I think from 2010, we are into the pharmacovigilance programs and all over. Almost we have 15 years, we have come all the way. And definitely it is progressing to a level where I think it cannot be ignored. The program, and as well as the ingredients of it. So as far as the PVPI, what we call Pharmacovigilance Program of India, is a crucial for identifying and analyzing adverse drug medications effects, and even those may be rare, it can be also be a very delayed one. I think even that also has to be really captured. So that is where I think probably a very notable example. I think I can keep on giving some of those examples, as we all know that in some of those carbamazepine thing which has been there for the South Asian population, which was not detected in earlier, but only at a very specific population because of the genetic reasons and all. So that would have been that was actually could able to captured only through this PVP program. So I think even the CDSCO, which is giving those approvals and all, the data collected through PVPI, pharmacovigilance programs and all, ultimately they will be a predictable decisions can be informed decisions can be taken by while giving the approvals and all. And also after even issuing the approvals, and also there could be some changes with respect to the labeling. You can also modify, and you can also suggest some of those measures. And above all these things, if it is not really helping, and there is a very important adverse reaction has happened and all, and I think the regulatory will not hesitate to withdraw that from the market. I think these are some of the things which will be really helping them. So ultimately, it ensures that only drugs, as I said, that which only has to benefit the patient rather than it harm it. And we can also create the cultural of safety in terms of promoting the culture of vigilance by encouraging healthcare professionals and patients to report ADRs. I think here we have missed only talking about the healthcare professionals and about the patients. I think there is another link also is the pharmacist. A pharmacist at the pharmacy levels, where he is one of the important link between the patient and the prescriber, and the patient always try to engage with him, and he is the one who can able to pick it up very early if there is something very adverse, or even a very minor changes, also minor issues that have been detected, are observed by the patient, and has brought to the notice of the pharmacist at the pharmacy level. We have around 10 lakh pharmacies in this country. I think that is where one of the important link. So these are one of the important aspects of the entire program and all. And as we all know that we are the pharmacy of the world, I think we still have to build that trust and confidence in our manufactured, the Indian manufactured drugs, not only domestically but also across the globe. I think that is where I think we are able to bring this program together with the Global Pharmacy Pharmacovigilance Week, and able to bring that kind of a trust. As we try to maintain that uh, we don't mind what you call a trade deficit, but not a trust deficit. That kind of a that kind of a tagline we go with that, and you can see that the compliance and commercial things both are actually being highlighted in this program from our programs actually. So as we all know that our USP of our products is not only quality, affordability, but scalability. And I think probably we can add one more dimension into that, that you can trust the pharmacovigilance program of our country also, where we can able to give you those kind of signals which has happened in this country, and to make them understand that the drugs which are being produced are safe and efficacious. I think as I have tried to bring some of those points into this one, the global mindset we should always be there, that though we are different countries, different regulations, but the universal concept goal is only that the patient protection. That is what I try to mention, that in a cardinal principle of that one. And the preparedness with respect to our inspections and audits are about all these are reflections of our own cultural of compliance. So I think probably I will not take much of those timings and all, but important is that while trying to detect all these things, I think probably while coordinating with all those in the stakeholders of this program and all, the important point, as I rightly mentioned, that now the present the digital tools, I think embracing them, the AI, automation, and the data science will definitely allow us to predict and prevent risks more effectively than it was earlier. So, and also that respect to transparency and engagement. I think this is very important, and we should say that the patients are not just subjects. In fact, they are the partners. We have to encourage them to tell, to express, to bring it to the fore about the issues which they have encountered. And there should be and there should be a platform, or there should be information feedback from them, either from the patient or from their attendants, or who are the caregivers, or whoever it may be. So these are all very important in that sense. So let us recognize and applaud the dedication of every pharmacovigilance professional, and we can try to build that kind of a platform where the patients and everybody can try to report those issues in terms of the drug safety, about the drug safety and all. So I think not taking much of the time, I think the eminent speakers are in line with that. And thank you so much for bringing me, making me a part of this Global Pharmacovigilance Week. And I wish you all a meaningful and inspiring Pharmacovigilance Week. Thank you so much, Majiddin. And also, I think the Industrial Pharmacy Division of IPA, in fact, I think kudos to you, and making this happen for all of us. Thank you so much.
Thank you, sir. You inspired us so much with your words. It's always welcome to know what is our vision and what exactly is being expected from Indian pharmaceutical market and also manufacturers. Thank you, sir.
Thank you so
It's
much.
a great honor to have you, sir, here. Since you have given a vibrant speech
Thank you,
where,
Sridhar.
yeah,
Thank you,
entire
Sridhar, for the opportunity.
Yeah, thank you, sir. And it means you have given the complete insights on the predictive on the India pharma into the entire globe, how we are moving ahead.
Yes.
So it's a great honor to have you in this forum, and to the viewers, the correct message has went because of the Patient Safety Week,
Yeah.
and we are very delighted to have you here, sir.
Yeah, great. I think it's not, that's what I said, and it's not just commercial. I think compliance and also is important for us. So we are.
Correct.
Thank you
Correct.
so much. Thank you.
And I apologize for because I was
No,
in
no,
the
no, no.
other meeting.
Yeah.
And Dr. Milind is also actually is traveling because of the IPC Congress related meetings, but he will be
Yeah.
joining some other time in this 1.5 hour.
Sure. I'll be
Yeah,
happy
but
to.
yes, sir. So and
But
we will.
if I have another meeting, I'll join, and if try to juggle between these two.
Sure,
Yeah.
sure.
Thank
That
you
will be
so
good,
much.
sir.
Thank you.
Yeah.
I think all the best to Dr. Shraddha ji.
Thank you.
I think that I'll be listening to you.
Thank
Thank
you
you,
so
sir.
much.
Thank you, Sridhar sir. Thank you, Rajapani sir. Without any delay, I'll go ahead with the next speaker. We have wonderful dynamic speaker, Dr. Shraddha, Senior Medical Safety Lead, Patient Safety from Sandoz. Dr. Shraddha is a Senior Medical Safety Lead in Sandoz. In her
No, correction, Mujib.
yeah.
It's not Sanders, it's Sanofi, correct?
Oh, okay. Sorry. I think I have taken from.
No, it is Sandoz.
Oh, okay.
Oh, sorry. Okay,
No
Okay.
good.
worries. No
Yeah,
worries.
thank you.
We all work for patient safety, so
Yeah, yeah,
that's okay.
yeah. So in her current role, she is involved safety of products such as signal detection, literature review, and development of core data sheets and responses in inquiry from regulatory authorities or healthcare professionals on safety issues, risk management plan. Prior to this role, Shraddha was medical director for Eversena for 5 years. There, she was responsible for leading and delivering and implementing end-to-end pharmacovigilance services, especially in signal detection and risk management for small medium-sized pharma companies and also different therapeutic areas, including US and Europe markets. She was also leading business development and training academies. Overall, Shraddha has more than 16 years of PV experience in pharma and CRO. She has worked in Paris for AV Sciences, a French biopharma, as deputy QPPV. She has worked in Oakheart Pharma as a pharmacovigilance manager. Prior to this, pharmacovigilance expert with Novartis Healthcare in India, and also senior medical reviewer with Cognizant Technology, a leader with strong management strategies, development, and business development experiences, underpinned by extensive domain expertise. Shraddha obtained her master's in biomedical technology at Coventry University, United Kingdom, also MBBS from in medicine from MUHS University, Nasik in India. And she has also done certifications and diploma in genetics, Six Sigma leadership topics, and she has authored a pharmacovigilance module in Symbiosis Institute for diploma in clinical research. Without any delay, it was a very big introduction, Dr. Shraddha, but thank you. So without any delay, let's listen to our next speaker. Laukik, can you share the slides, please? Laukik, can you share the slides, please? Over to
Yes, sir.
you, Dr. Shraddha.
Thank you so much for that extensive introduction. Now I'm feeling more anxious than I was. But I mean, I'm really humbled by the invitation, and Dr. Sridhar has been very kind to me to involve me in such kind of great initiative. Patient safety is heart of everything that I do, and I'm very passionate about it. But it cannot be done in silos, as Dr. Raju Bhanu was saying earlier. And I'm very thankful for Dr. Sridhar that he has invited me for this very interesting webinar series you guys have put in together. I know it must have not been easy to coordinate and collaborate with a lot of industry experts, academicians, and everybody else who is passionate about patient safety. But of course we do have a lot of meetings and stuff, but you did not give up and you have put together a 7-day series of webinar. That's really commendable. So I'm really honored to be here, and I'm really grateful for the opportunity to talk about pharmacovigilance and patient safety. Okay, so I know I have taken a lot of time to thank you, but just wanted to take time to appreciate the core principle that you are trying to bring in, which is raise awareness about patient safety and also professional learnings together. So thank you very much. And without further ado, I would like to start my presentation. The topic is benefit risk assessment, and it's really a key topic because it's not an individual topic that we can address like a PSUR or an RMP, but it's a heart of everything that we do as a pharmacovigilance professionals. Can we go to the next slide, please? Okay, next slide. Yep. Okay, so what I would like to cover in this, and you can, click because the animations will take time. So to understand the benefit risk concept, then what are the methods and sources we use in BR assessment? What is the use of BR in pharmacovigilance? And probably if we still have some time left, I would like to see how do we write a benefit risk assessment, or how do we come to a conclusion of BR assessment when we follow a particular document or a particular concept in pharmacovigilance. And before we begin, can we go to the next slide, please? Before we begin, a standard disclaimer: all of these opinions and the presentation belongs to me, and these are my personal opinions. Sandoz does not, those are not those of Sandoz. So and then we start with the topic. So key concepts before we go into the discussion on how do we do the benefit risk assessment in pharmacovigilance. Let's first understand what is benefit, right? So we use benefit and risk in our day-to-day life very frequently, and even though we know what it means, but in pharmacovigilance they mean different things, and it's important to put that in context before we go into the granular scientific details. So benefit is something that adds to the that has clinically meaningful improvement to a patient, whether it's impacts their quality of life or whether it treats or prevents the disease. But that's what benefit is. And when we talk about risk, as I said, we use term risk in everything, and that goes for pharmacovigilance as well. So risk could mean anything, and hence it is very important to put it in the in the context. So risk could be an adverse event. When we are talking about an ICSR, we simply use the term risk. But when I am talking about risk and benefit, it's a very key concept to know that the risk in this context mean that the impact of that particular risk on the patient with respect to the disease that they have, and the quality of their life, or the complications of the disease, they could be worsened because of the drug. So we are talking not only about the patient, but the impact about the public health impact of that particular drug. So it's very important at an aggregate level to understand that when we talk about benefit risk assessment, we are talking about the benefit and risk assessment of that particular drug on that patient, and furthermore on the impact on the public health impact. Next, please. So the integrated benefit risk evaluation is performed for all the authorized indications. And I really like this table that I have taken from the EMA's fourfold model, and I have provided the reference here, wherein we talk about what are the favorable effects and unfavorable effects. And it's very logical. I don't need to spell out the definition, what means favorable and what means unfavorable. But what I wanted to show through this diagram is that both of them have uncertainties. So when we are talking about risk, we have to remember it's a potential risk. There are uncertainty or uncertainty of those unfavorable effect which they will occur in the patient or not, and hence it is important to put that in the context when we are talking about the patient safety for that particular drug. Next, please. So why benefit risk assessment is important, right? So let's take an example. I have taken this as a blinded example. If we have product A, which offers you one benefit and one risk, and versus we have product B, which has one benefit but two risks. So logically, when we don't have any information, we think that product A is better than product B because it has one benefit and one risk. I wish our real life was as simple as that, and then pharmacovigilance professionals could go on vacations. But that's not the truth. So, what happens in a real life scenario is, if we can go to the next slide, we'll see the real example. Right. So let's say that now I give you more context. So the product is one which is used in an indication for infection, and this infection is wherein if left untreated it leads to life-threatening sepsis. So now you have product A, which lowers the risk of life-threatening infection, but it has a risk of reversible mild neutropenia. And then we have product B, which treats the infection, but it has serious neutropenia, which is reversible, and and mild liver function enzymes are increased, but it is reversible and treatable. So now when we put all of this in context, we know that the benefit of the product B outweighs the risk, because it is treating the infection, which if left untreated would have caused a life-threatening sepsis in the patient. So we are accepting the risks because we can mitigate those risks. We see that the neutropenia is serious, but it is reversible. The liver function enzymes are increased, but they are mild and they are reversible and they are treatable. So when we put this in the context, we are able to conclude the benefit risk assessment of product B is better than the product A. Let's go to the next slide, please. So where do we need benefit risk assessment, right? We need it in your PSURs. We need it in your risk management plans during your marketing authorization application, even for the renewal of marketing authorization. And we also need to do a benefit risk assessment when you are updating your labels, or when even you are preparing your patient alert cards or HCP educational materials, things like that. Because at the core of everything that we do in pharmacovigilance, we have to ensure that the benefit outweighs the risk, and that communication has to reach the clinicians and the patients as well. So there are different ways that this information that you have concluded about your drug then gets segregated into different documents in the pharmacovigilance. So these are some examples that I have put on the screen where we need to do the benefit risk assessment. So let's look at what are the components of benefit risk assessment, right? So the first is disease indication. Um, what is the underlying disease that we are thinking, that we are targeting for that particular disease? So for example, pathophysiology of that disease, what is the epidemiology, what are the risk factors, what are the complications, and especially we need to also pay particular attention to what are the therapy goals. So the current clinicians or the HCPs, what are they targeting when they are prescribing or administrating this drug? So that we have to consider when we are thinking about that particular disease. For example, we all know Zolengensma, I hope I'm pronouncing it right, is the world's costliest drug, and it is being used for SMA. Although it has a very severe safety concerns, it is still being used because the indication in itself is it is used as a one-time gene therapy for a fatal pediatric disease. So here clearly the benefit outweighs the risk. So we have to understand what the underlying disease indication in itself is. When we talk about epidemiology, is the patient population a vulnerable patient population, pediatric or geriatric patient population, or patients with kidney disease, cardiovascular comorbidities? So in those particular patients, we have to think about unmet medical need, and that's when it is important that one of the key component of your benefit risk assessment is to understand the disease itself, and what happens if the disease is not treated, what kind of complications we are looking at if left untreated. Risk factors, what are the factors that increases the probability of people getting that particular disease? So, those are very important to then understand and put together a benefit risk assessment wherein you compare the patient population versus those who don't have that disease, and then you are able to understand if any particular adverse event occurred in a particular patient, you would know if you would it would be easier to do a causal association whether it was caused by the drug or not. So it's important to understand that one of the key pillars for any benefit risk assessment is the disease itself. Then the need of the drug, what are the current treatment options available? As I said earlier, I gave you an example. There are many diseases like autoimmune diseases where the only treatment available or the treatment modalities available are palliative, or they are symptomatic treatments. For example, corticosteroids for autoimmune conditions, but they do come with their own challenges. They have to be administered on an ongoing basis. They have their own safety concerns when used in a long term. So if the current treatment options are palliative or symptomatic, or they have severe safety concerns, then we need to think about what this new drug or this new drug would be offering the patients. Is it going to prevent the disease? Is it going to treat the disease? And even if if it is not going to prevent or treat the disease, is it better in terms of the safety profile when compared to the current treatment option? What are the specific advantage of the drug that is going to offer against the current options? Then the third key pillar is the benefits. As I said earlier, benefits are the key clinical outcomes that will improve the patient's quality of life or the patient outcomes. And then the risks, these are the key risks that will have an impact on the morbidity and mortality of the patients. And we are going to say the key risks that we consider during benefit risk evaluation are those that are reversible and non-treatable. Finally, when we have looked at all these four pillars, then the conclusion could be, is the benefit risk profile positive, which means benefit outweighs the risks. Let's go to the next slide. So as I said earlier, right, so one of the key component of benefit risk assessment is to look at the disease and the patient population that we are going to treat. So when I said disease and the patient population is especially pay attention to vulnerable population, if they already have comorbidities, any risk factors, and what are the current therapies available in the market? Is there an unmet need? We all remember we had COVID-19 vaccine during COVID-19, which was accelerated approval pathway because there was an unmet need in the market. We were in a middle of a pandemic, and although the long-term safety concerns or the long-term safety data was not yet available, still the vaccine was made available to the patients, to the population to prevent the complicated COVID-19 complications that the disease in itself was going to cause to the patients. So need. The other example could be Trikafta, which is used, and the access is not so easy. However, it's used for a patient with cystic fibrosis mutation, and it's the only drug that is available for these patients. So there is clearly an unmet need, and hence, although it has serious safety concerns of liver injury, it is still being approved to be used in this indication because there is an unmet medical need. Then the third component is the lower risks to what the current treatment modalities are available for the patients. And the fourth component is the risks. What are the risks that the drug X or drug A that you are evaluating has? As our chief guest was saying earlier, we don't like surprises. Regulator don't like it. Manufacturers don't like it. The only time we like surprise is when it's our birthday. That's why it is important and critical to know what are the risks for that particular drug, and to consider them. Are they serious? Are they severe? Are they treatable? Are they preventable? And we will cover that in subsequent slide, because what is key is, once you know the risk, you know how to mitigate them. You have risk prevention measures or risk mitigation strategies to then prevent this risk, and to ensure that the benefit risk profile of the drug remains positive. Can we go to the next slide, please? So what are the components of the benefit risk assessment? And as I said, to understand that, we have to understand the drug. So when we talk about the drug for which you are doing the benefit risk assessment, you have to consider whether this drug is going to cure the disease, or whether is going to prevent the disease. Is it a disease modifying agent, or is it going to be used in a life threatening and life prolonging situations, and whether it's just a symptomatic treatment for the disease. These are very key components to consider when you are doing a benefit risk assessment. Can we go to the next slide, please? So, and when we talked about the product, the same way we talked about the product, the same way we have to consider some key components when we talk about the risk. So the risks that are seen with your product for which you are doing the benefit risk assessment, you have to consider whether this particular risk is serious, is it severe, and especially once you know that, you would like to now think about how do you prevent this risk, how do we mitigate this risk. Is it treatable? Is it something as simple as rash? If the patient is just going to get rash and the drug is a life-saving medication for some disease, you can treat it easily with some topical steroids or topical ointments. Is it preventable? Is it what is the frequency of this risk that can occur in a patient? So these are some key components to consider, because once you know that there is this risk that can happen, that there is this potential risk that can occur in your patient, then you can think about some risk mitigation strategies to then balance the benefit risk profile of that product. Can we go to the next slide, please? So what are the data sources that you can use when you are doing benefit risk assessment? There's a huge data sources, and then there are multiple methods that you can do to do the benefit risk assessment. When you do a benefit risk assessment, trust me, it's not easy, because you are really covering all the aspects of that particular disease and that indication. So you sit on a huge pile of data, and then you make a meaning out of it. You build a story for your product, wherein your product is the hero of that story. And then you need to have an evidence to say that product A has a benefit risk profile which is positive. So what is that evidence? The evidence you look in is the epidemiological data, your spontaneous reports, your clinical trials, non-clinical data, real world evidence, real world data. So these are some of the data sources and methods, depending upon whether your drug is new and unapproved, or whether it's a known, whether it's an approved product, but you are thinking about extension of any indication. So those are some data sources that you would consider when you are doing a benefit risk assessment for your product. Can you click on the next item? Okay. So there are some methods that could be used which are quantitative, and then there are some that can be used qualitative. It doesn't matter which method you use. What matters is to build that story. Even the regulators would like to make sense of the data that you are presenting, and to clearly define what method you have used for them to put the context to your story. So depending upon the level of the evidence you have and the stage of the product development, you can choose any method you want. As I said, it has to make sense to the regulator, or to the reader what you're talking about, because ultimately what you are going to try to say is that the benefits outweigh the risk. But here is the evidence, and how you present that evidence is also critically important, and hence choosing the right method and right data sources is also critical. For example, if it is a new and unapproved product, clinical data makes sense. But however, if it is an approved product, what makes sense is your literature, your real world evidence, real world data, and all those spontaneous reports do carry a less advantage. However, if you do have some clean data from your spontaneous source, you can still use it as an evidence to then put your benefit risk assessment together. Can we go to the next slide, please? So what is the strength of evidence for benefit risk assessment, right? So one is a large database. So if your drug is being used in a particular indication, and you have a large volumes of data to then extrapolate what kind of a benefits or what kind of a risk that a drug offers. Uh, However, it comes with its own disadvantages, because large data means a lot of noise, and then putting it in a consistent manner and making sure that the data does provide a good understandable evidence makes it more um, is more important. And then the most important data that provides a very good strength of evidence is randomized control trials. And especially when you're putting this story together, it is important that you have data that has clearly defined clinical outcomes. So if you have looking at a drug for asthma, let's say, suppose, so then you have to have a clearly defined clinical outcomes, whether the drug lowers the episodes or whether the drug is just providing a symptomatic relief. What is the clinical outcome that you are looking at? So it's very important that the data that you are choosing, or the evidence that you are choosing to be used for your benefit risk assessment, is clearly defined, is consistent, and it provides the context to what you are going to ultimately say. Then and throughout this benefit risk assessment, you have to ensure that the evidence that you have provided can be used to then build a causal association with clear confounders. Whatever you have put in that a drug A causes risk B, what are the confounders? When does this risk B occur in that particular patient population? When the patient has underlying autoimmune disease, when the patient is on any other concomitant medication. So what are those confounders? You have to put together a context to the story that you have built. Can we go to the next slide, please? So when I talked about, as I said earlier, the strength of the evidence is important, and so we have to choose the right method and the right source of data or the evidence. There are, even though if you have randomized clinical trials and a very good data, there are still some uncertainties or unknowns that will still remain in benefit risk assessment, which is lack of long-term data, confounded data, and small study population or observational studies. um, in this case, again we go back to the basic, to focus on unmet need. So if your disease in itself was a rare disease or an orphan indication, then you will not have a large data. In that situation, you have to focus on what kind of a data and method you will use that makes sense and will provide an evidence to say whether the benefit risk assessment was positive or not. Can we go to the next slide, please? Yeah. So then you have then you have some documents which I have put together to just give a context to what is expected in this document when you are doing benefit risk assessment. One of it is clinical overview, and this document is a part of your MA application, and herein you are required to provide your benefit risk assessment and interpretation of the efficacy and safety of the study product that you are presenting. Can we go to the next slide, please? In the interest of time, I think, yeah. This is a very good table that I have taken from FDA's benefit risk integrated assessment document, and it provides a very comprehensive and easy to understand benefit risk evaluation, how it should be done, wherein they have put in some dimensions what you need to consider, and then the evidence and uncertainties on the other side, and what is your conclusion and reason. As I said, analysis of the condition, the 4 pillars: disease, drug, risks and benefits. So what is the disease? What are the what are the uncertainties in the diseases? What are the current treatment options? The unmet need? What are the benefits? Your primary and secondary endpoints, if you are talking about a study, or your primary and secondary outcomes when you are treating those patient populations, and then what are the risks, and how will you mitigate those risks? So this is a very good comprehensive table that you can use to build your benefit risk assessment and to provide the evidence on the conclusion that you have concluded the benefit risk profile of the product is positive. Can we go to the next slide, please? The other critical document that we do, and which is a bread and butter in our daily life as a pharmacovigilance professionals, is the periodic benefit risk evaluation report, or PSUR. And we do an integrated benefit risk assessment in section 18.1 and 18.2. However, the evidence, this is a conclusion section. The other sections prior to section 18 are the ones where you provide the evidence, and herein you are just concluding why the benefit risk of that particular product is positive. But the evidence is provided in the earlier section, and the concept more or less remains the same. The benefits should outweigh the risks. So if you are talking about the benefits of the drug, that it treats the particular disease, or it is the only drug available for that particular disease, but it does come with the risks. And what are those risks, and how do we mitigate those risks, or how do we manage those risks? So that is how you will do in the periodic benefit risk evaluation report. The other document that is common in pharmacovigilance is the risk management plan, wherein you will also do the benefit risk evaluation, and you will provide the conclusion. If you can go to the next slide, please. Yes, can you click? As I said, we have covered these concepts wherein we have to think about medical need, wherein you think about the disease, how the disease patient population is, what is the duration of the disease, how severe it is, how serious it is, if left untreated, what could be the complications, and what are the alternative therapies available, and then how this particular drug that you are writing the PSUR for offers the benefits, and what kind of benefit it does often. So the benefit section is 17, but the conclusion is in section 18, when you put the context of the benefit in term along with the risks. And then the risks are they reversible, are they treatable, serious, severe, and the details of those is provided in section 18, section 16, but you do conclude in section 18, wherein then you say that the benefit risk profile of the product remains positive. You can also talk about the strength and limitation and uncertainties of the evidence that you have provided for both the benefits and risks, so that the reader of the document understands why you say the benefit and risk remains positive based on what evidence. Can we go to the next slide, please? I have provided some screenshot from the PSUR or PBRED template, and we have covered the concept on the earlier slide. Only the difference is that the section number is different. However, the key concept remains the same. So I think we can skip this slide and go to the next one. So as I was saying earlier, another document that is part of pharmacovigilance professional's life or a daily bread and butter is the risk management plan. And as it says itself, right, it's a risk management plan. So it's important to do a benefit risk assessment when you are doing risk management plan, and not just focus on the risk, because you have to bring in the concept of benefit when you are doing the benefit risk assessment for that particular product. Although the RMP has its own template and the focus is only on the risk and how we will mitigate those risks, however, at the back of the mind of a pharmacovigilance professional, you have to remember to also consider the benefit as a key concept when you are presenting your risks. We can go to the next slide, please. Yeah, so I have put some context and some things on the screen share, but I don't want to go into details of this. It's a very standard template. What I wanted to bring to the attention to the audience is that benefit risk assessment is not some key document that you do in a silo, or that you do at the beginning of the product development and then it's done. It's an ongoing activity that as a pharmacovigilance scientist you have to keep doing it through various compliance regulatory required document. But however, you have to also ensure that your ongoing pharmacovigilance system allows you to keep on thinking about what are the benefits and what are the risks, and to ensure that the benefit risk profile remains positive. Can we go to the next slide, please? So, in a nutshell, what is benefit risk assessment? Why we do it? It simplifies products understanding. It is utilized for multiple pharmacovigilance document, it makes an impact on the patient safety, and it adds credibility to your product or to your molecule. What is it? We saw the 4 key components of benefit risk assessment. One is the disease, the other is a drug, the third one is the benefits and the risk. And putting all of this together in a context where you provide the evidence for each of these pillars is critically important. How we do it, we have to choose the source and the method depending upon the disease or the patient population and the drug. And then you have to provide a concise and a clear, transparent analysis, what method was used and what source of information was used, so that the reader or the regulators understand on what basis you are saying the benefit risk as a profile of the product is positive. When do we do it? When you are beginning the development or proof of concept. It is done in a collaboration with pharmacovigilance or patient safety, clinical, regulatory, and medical affairs as a function. It's not a standalone assessment or a one document that can be done in silo. It is a collaborative effort by everybody from regulatory, clinical, medical affairs, and pharmacovigilance. Ultimately, I think what this our what we talked about earlier at the session is, it's important to have patient safety and patient centricity at the heart of everything we do. And that one way to do it is to keep benefit risk assessment as a key concept in our mind, so that we are proactive, predictive, and preventive in ensuring that the safety of the patient is at the heart of everything. I think this is my last slide. Thank you. I'll take a break here.
Yeah, thank you, Shraddha. Thank you for giving a very elaborative, all kind of documents. We have the next speakers. In the interest of time, I'll take the questions and I'll send it to you on a mail.
Sure.
Thank you, Dr. Shraddha.
I'll have one more time the next speaker. We have one more speaker.
Yeah, very lucid presentation. Kudos to you, ma'am.
Thank you very much.
Yeah.
Thank you.
Thank you, sir. Thank you for listening to us.
I'm a pharmacologist. I can't afford to miss that.
Okay, so I'm having my next speaker. Give me 1 minute.
Good evening, Dr. Avinash.
Hi, good evening. Let me.
Laukik, by any chance, can you share the screen for the introduction slide? My system is having some trouble sometime.
Yes, sir. I will share, sir.
Thank you. We'll appreciate.
Should I share my screen?
I have an introduction.
Okay.
Avinash.
Just a brief then you can
all floor is yours. So I think my slides have stopped working. I don't know, something went wrong with slides. Technology sometimes is very, very cruel.
I can self-introduce myself. It need not be elaborated.
Yeah, I think we have the slides on. Yeah.
Who's going to read all?
Can you make it as full screen, Laukik?
I'll keep it very, very brief, guys.
No, give the honor to Mujib, sir.
But don't keep it so lengthy. It looks like I am 100+ year old. This so lengthy introduction. Keep it very brief here.
Yeah.
Go ahead, Mujib.
Yeah. So Dr. Avinash Ramchandra Kakde. Okay.
Guys, I will introduce Mujib, let me introduce myself. This is too lengthy, too boring, and really.
Okay, sir. I'll park it here because I know that you wanted to jump into the session. You can introduce, sir. Should we stop sharing?
Yes, please. Yes, please.
Laukik, stop sharing. Sir wanted to take introduction and also the session also.
Good evening, guys. Good evening. This is Dr. Avinash Kakle. I am the global head of pharmacovigilance for Dr. Reddy's. By training and education, I am a physician, and would love to remain a physician to the core. My rest all biostats and all these courses I did were just an enabler to the work that I do and to the patients that we all serve. This is close to 19th year in pharmacovigilance in industry, and prior to that, I was a practicing physician. So thank you, pleased to be here, and thanks, Dr. Sridhar, to have me on board. I really did not get good enough of time when I was seeing Dr. Shraddha's slides, and I was like, oh, I will wind up in 20-25 minutes. I have just 12-13 slides. So excuse me for that, but I'll try to keep it very high level. I don't know the nature of the audience. Are those freshers or established professionals? This is definitely not for the ones who are in the industry for 5+ years. This is more of an.
We have multiple people who have got registered, Dr. Avinash Kakre, sir.
So I'm
We
trying to
have
keep
so
it
many people
very,
who are
very,
watching in
very,
YouTube.
yeah, very simple, very high level. We'll try to share my screen. Let me
Yeah.
know when.
Well, it's great honor to have you here, sir. Out of
Yeah,
your
thank
busy
you, sir.
schedule,
Thank
you
you.
have given time for
Thank you.
both
Thank
the
you.
speakers. We are very thankful for you.
It says sharing is not turned on.
Laukik, can you make co-host?
Okay, let me know. Or should I quickly?
No. I think now you'll be able to share the screen, sir.
Wow.
Laukik is making you co-host. Laukik, co-host rights have been given.
Yes, sir, it's given.
Yeah. So now I think you can share.
Okay.
Yeah, now your screen is on. Perfect.
Yes, you can see.
Okay, great. So just 14-15 slides. Let me know when you can see my. Okay.
Yes, we
Can
can
you
see, sir.
confirm? Yes. So this is a safe harbor statement. I will skip it. High level topics, but really I didn't get enough time to build the whole slide. And this is typically the definition of signal. And for those who are really 1 or 2 years in the industry, I will try to give you an anecdote of why even I believe signaling is the most important aspect of PV, and why over the last few years I really do not believe that signal is the only important aspect of uh, the whole end-to-end pharmacovigilance process. And by the end of the session, you will realize why I am making this paradoxical statement as to why I thought initially that learning signal management, specifically signal detection, will be sort of climbing Mount Everest. But why I believe now, and all those who have known me or worked with me may understand why I do not really believe that signal detection or knowing signaling is the only equivalent to scaling Mount Everest. So basically, coming to the definition of signaling, it is some new potentially causal association, right? And the words are very, English is a funny language. We all know that, right? For all those who know Amitabh Bachchan taught us even before I was born that English is a very funny language. And then the way we word or define the terms, whether it is a legal verbiage or a academic verbiage, we try to build in many riders. And so too for the definition here. It says new potential causal association. So the word potential itself acts as a rider that it may or may not be causally associated. And we all know that a temporal relationship does not really mean a causal association. The second part of this signal definition, any new information about a already known association between an intervention and an event or a set of related events. So now there are two parts. One, which talked about a new potential causal association. Some would also say that it could be a new potential association, and then we will see whether it was causally associated or not. And the second part is you already know an association, but you don't know the frequency, or you know the frequency, and now you see an increase in a frequency, or you see some nuances of that known association. Like for an example, drug A leading to, just keep it simple, leading to rash, a widespread rash. So rash is listed in the label. Okay. So you know that there is some association. Now, whether it is an AE, adverse event, or an adverse drug reaction, that's, we are not complicating our lives, but it is known, as to be listed with that drug. Now, what could be the new aspect? It was commonly listed as under the common frequency, or it is now we are seeing a rise or increase in the frequency, or you are seeing this rash only in patients who are having hepatic failure or renal failure, or who are taking this drug with some another drug, or is seen only in pregnant patients, or you are seeing this only when you take the dose, at a certain dosage, or you see that this rash is now seen only after third day or fourth day, or you are seeing immediately after a few hours. So there is a new aspect that you are trying to discover, or you are thrown into your face, that you see that there is some new aspect of this already listed rash. And then the bigger part is that this new association, or a new aspect of a known association, is good enough to take further action. Now, what is the further action is what we will see in the slides, right? Now, what is signal? Signal we have seen, the first bullet here is seen. Now, what is a newly identified signal? Something that came up during the reporting interval. Dr. Shraddha took a very detailed, I missed the first half an hour, but she took us through the PSUR, the PBRS, the reporting intervals, I presume. And so if you see something during the prior to, for the first time during that reporting interval, either of a safety aggregate report or at the safety signaling activity. So like the aggregate reports, we do have a data log point for signaling activity as well. And if during that reporting interval, if you see the signal for the first time, that's a newly identified signal. Ongoing signal is something that you have seen and you have decided in the last cycle, where with that you have to continue assessing it, monitoring it. Okay, and is still not, the whole process is still not complete at the time of the data log point. The potential signal and signal, there are multiple school of thoughts depending on how your SOP talks about. Someone would say that signal is as good as a potential signal, and someone would say it's very good to carve out a specific term potential signal. But I believe that a signal by in itself means a potential signal. Now, refuted signal is something that you have done some verificatory action and concluded that as of now, you don't see that there is some meat or some real association that can be determined. And that last few words at that point of time is very important, because life has a funny way to prove us wrong. So maybe 2 or 3 years later, that same a drug event pair may become more may get more evidence, and you would like to revisit that that it looks like there is a some further evaluation that is required. Now, where does signal management or signal detection come into picture? At the start, if you see the whole life cycle of how the adverse event goes through, and that's a circle, right? So I don't know where to start, but let's assume that you start off with the launch of the product, commercial product, right? And that let's take that as a starting point. And you say that uh, you have launch a product in the market. Uh, you get a call from a consumer uh, through your toll-free number, and you get to know that there is an adverse event. And now, depending on whether it is a valid case, invalid case, you database the adverse event, basis the GBP6 and your local SOPs, you process the case. Okay, determine whether it meets the expedited or periodic reporting, and decide on the whole nitty-gritties of case processing, the data entry, quality review, medical review, and then do perform the expedited or the periodic submission. And for that time point, you have closed that case, by raising adequate follow-ups for some deficient information. So now as far as that case is concerned, that ICSR, you are done and dusted with that. You do a lot of AE processing over the year, and then you see that you have a good amount of data that is generated in your database. Similarly, you do a lot of literature surveillance activities for the said product, and you have generated a good amount of data. So at the end of the say let's take a 6-month time period. You have a good amount of data that has come in through multiple sources. One is the call center, one is the literature that I spoke about, the third could be a social media surveillance, the fourth could be uh, someone writing to you to your mailboxes and then gathering some information, someone reaching out to directly to the quality team, and you have something that is a mixture of PQCs and a product quality complaint and an adverse event. The fourth could be something that you are observing, you are doing an observational study, phase 4 study, and then you are generating some data from there. Then the fifth could be you are seeing, huge data sets, uh, publicly available or subscribed databases that your company has access to. So you have a full blown set of data in your database. Now, what do you do? What do the regulators want you to do? ICSRs, you have sent it in a real time, either as a expedited or a periodic, and then club it with the periodic aggregate safety data, safety report. In addition to that, the most of the agencies do mandate that you have to mine the data that you have to see if you discover something which is meeting the definition of a signal, where you can identify a new association or a causal association, or you see that there is an increase in the frequency of a known association. Now, how do you do that? For you to do that, either you do it in a very primitive format, like how it was done, 2 decades ago, where you use the SAS and then pull out the ratios through the SAS, or even in the very first company, long time back, we used to do it very primitively on Excel, and that was the funnier part of the whole uh, exercise. But now almost 1.5 to, I would even say, more than 1.5 decades, you have validated signal detection tools where you have to just import your safety database data, and it will cull and mine the ratios that you have pre-configured, and you get a signal of disproportional reporting. It's not in the slide at all. I'm just talking. So we create SDRs, right? Now, there could be various ratios that your company agrees to do, and there is nothing called as absolute right, absolute wrong. Nothing ever in life is absolute right, absolute wrong. Doesn't the same applies to signaling as well. So your company must have chosen, 2 or 3 ratios, and then using that ratio, the set of ratios, you will get some output on what the database believes to be a potential signal of disproportional reporting. So you have something that has come up. So all these, thousands and thousands of cases that you have, and then you get something meaningful, not fully meaningful, some meaningful data that is thrown out. We call this exercise as quantitative signal detection. So you are using the databases, applying the statistical ratios. It could be combination of n equal to so and so, PRR equal to so and so, chi-square equal to so and so, or you have a different set where you use EB05 as one of the component, you have PRR as so and so, n equal to so and so. Then you do that mining in terms of the time frame for the reporting, the interval data, as well as the cumulative data. So there are multiple ways of doing it, and I don't want to really bore each one of you on giving what is good and what is not good. At the end of the day, what is important is what are you going to do with the data that is thrown out of this quantitative signal detection. Doesn't make any sense when you only see the quantitative signal detection out. You have to do a qualitative signal detection on top of it to see, open each and every ICSR or a case that is thrown out to see if it really meets the definition of signal, right? Or a potential signal. Now that's how you start analyzing more. And that's when you have physicians kicking into the picture, where using not just the pharmacology knowledge of the drug, but also the knowledge of clinical medicine, where you try to make sense of each drug event pair that is thrown out from the quantitative part of the whole exercise. And then when you do this qualitative signal assessment is then where you get onto a meeting after the exercise is done, And that meeting is called by various different names. Few companies call it signal detection meeting, signal management meeting, whatsoever, reporting for the management of the company, whatsoever. And then you conclude which are the signals that you really need to refute, or you keep it ongoing, or you confirm that this is, we have to do some further action on whatever we have identified. So this is a very high level end-to-end process. Now I'll go through the slides. And another part of it is a subset, or I won't call it really a subset, but one that feeds into the signaling to a certain extent is a emerging safety issue. And this is a very, even the whole signal detection is defined by the European guidelines and very less so by the US FDA. So emerging safety issue is something that has a potential to cause a major impact, unlike signal, where you will have to really think through, go through the whole process as mentioned in GVP 9, GVP module 9, and its annexures. But emerging safety issue, where you say that I believe that this is something that will have or may have impact on the risk benefit balance. Dr. Shraddha took us through the how do you determine that risk benefit balance, or anything that has a wider impact on a patient's or public health, right? What is the biggest differentiator between signal and emerging safety issues is the fact that the emerging safety issues need a prompt regulatory action, versus signal can take its own sweet time. It will go through the minimum mandatory steps. You reach out to the agency will guide you, or you submit it through a PBRR, and then the prac will assess, come back to you. But ESI, the emerging safety issue, is something where you need to tell the agency right away at that real point of time, where you believe that the action by the agency would be a temporary restriction on the use of a medical product, or unfortunately, altogether revocation of the license to market. Now, these are the steps, and this is all there in the guidelines, right? Detection, the steps of signal management, signal detection, validation, confirmation, analysis and prioritization, assessment, and recommendation for action. And this is also a slide available. I can't see this, but then if I were to, can you see this now? Or so here, if I were to reduce this, you will see who is the owner of these steps. Can you see this now? Hello? Can someone confirm?
Yes, we can see.
Yes.
But maybe if we can put it on a full screen, it would be
Hold on. So
easier.
I have to move this.
Yes. This is better. Yeah.
So you have the first three steps, which are done. The first two steps are done by the marketing authorization holder, detection and validation. Confirmation onwards goes to the agency, and especially the ones in red, analysis, prioritization, action is done by prac, and then the exchange of information happens at the all the stakeholders are involved. So all that I said about qualitative, quantitative lies in these two steps, where you try to detect and validate a signal. Okay. Now this is also what I spoke about, that it is an exercise of identifying signals using any data from any source. It could be a spontaneous case, literature, it could be a post-marketing study that is ongoing on, it could be a social media, it could be a large data set like you have from the EMA, the EVIDAS, or the FAIRS database, or you have the database that Health Canada has. So you could have any data set that you are using for your pharmacovigilance activities. Now, as I earlier mentioned, it involves a quantitative as well as a qualitative review. Using the quantitative review happens using the statistical tools that we have. And it's always, most of the scenarios, it's a combination of both. Unless you are working on a niche product and you are the innovator of that product, and so the statistical won't really apply because you are the only one who is developing the product. Okay, and then as the molecule moves through various phases, you are only doing a qualitative assessment of all the data that is generated. So this is the detection part. Now, how do you validate whatever you have identified from the earlier step? Now, here you do this individual case at a MR level. You have a physician take a look at the DE pair or the data that is pulled out, and you try to make sense of what is presented to you. And you see from all the angles, and that is as good as a physician reviewing a ICSR. Now you have to bring in all the factors that you would consider while doing a causality assessment of an ICSR, of a drug event pair, basically. And then you justify or exclude that this is something that should be probed further, or this is something that as of now, you don't believe that it has sufficient information to label it as a signal as of now. And as I mentioned, it could, you could have to revisit your decision a couple of signal detection cycles later, because each one of us, all the MAH are limited by the amount of data that we have access to, whereas the regulator, has a wider data set to make the decisions on whether it is really a signal or not. And when I say it, the association or the drug event pair and the association that is identified has is like a signal or not, right? So this is where you validate. And here, when you do some validation, that's where the individual expertise kicks in. How much visibility or how much knowledge I have of the product, of the label, of the case, more so of the case, and putting all these 3-4 factors together, you will be able to make a very good analysis. And that's where having a high quality ICSR is important. Because if your ICSR doesn't have uh, a good quality, you will never be able to pick up something meaningful at the signaling step. If your ICSR doesn't have all the data, it is just 4 minimum criteria that is entered into the database, it won't give any insights into the case. And unless you have as much as data that is possible, you won't really be able to make a judicious, informed decision about, is this causal association worth exploring further? Should I call it as a potential signal or a signal, and then should I keep it open for further few signals, or should I refute it right? And that's why at the start of my talk, I did say that signaling is not that important. What is the what is most important and crucial is the ICSR process, the correctness of the ICSR, whether you are doing adequate follow-ups to the ICSR as required by the guidelines or by your SOP, is the quality review and the medical review done in a better way, or the best way available at your disposal? Do you have all the data, or has your ICSR team asked all the relevant follow-up questions that were required to be asked, so that you have all the data that a signal team would require at the end of the cycle or at the end of the period, so as to make a decision on the signaling? And that's why, having ICSR, I always say, is like a cell of a human body. It is the smallest living unit, but the most important living unit. The better your ICSR quality is, you could exponentially multiply the quality of signal detection or signal management exercise at your end. It is very important to have a end-to-end best ICSR that one could ever have to help the signaling part. This is all that. How do you validate whether you have the data into the SMPC, in the label, what was done, what is the strength of the evidence, what is the number of cases that you have? So this is all that I just spoke. Also, the additional sources, the second bullet here, is also about the same thing. See, I have covered by now in the half an hour, I have covered almost all that is written in the slides. Larger data sets, other agencies, what have they done with that signal, or have they reported a signal or a ESI? Uh, Everything I think I have spoken. I'll just run through the slides. Now this is from the GVP9, so it is also available, right? So when you validate, whether you validated a signal or not, if it is no, then you call it as a non-validated signal. And most of the time, the quality of ICSR is so bad that you have no other option to go back and then do a follow-up call to the consumer or the end user 6, 7, 10 months down the line. And one of the reasons why we see that uh, uh, we don't go anywhere when it comes to signaling is because of the incorrectness or inaccurate data of the ICSR. But if you have everything that is there and you tend to believe that the signal is validated, the flow here, then you decide what do you do with this, whether it is a new or a change risk. If it is no, then you say it is refuted, but you have a change to the frequency or altogether new association, then you propose actions such as changing the label, or adding some additional risk minimization. But a part of this is you cannot do at your own end independently. You have to consult the health authorities for sure. So this is what we just spoke. And if you remove this heading of the slide, it's all about a mixture of what you do at a ICSR level and at a PSUR level. The most important is the last bullet. So when you analyze a drug event pair in the light of signaling activity for a product A, and if your company has product C, which belongs to the same class of medicinal products as product A, there is a moral responsibility or a regulatory responsibility to apply the same association for product C and C and check whether is it particular to product A or is it also applicable to the similar class of products, like as in product C in this example. So that's one aspect of a signaling where you don't limit it to the product under consideration, but also apply it to the same class. This is the process flowchart. It's very much available in the GVP guidelines. It talks about timelines on what to do, where the image will come into. This color coding is basically on the responsibilities. It is a very, I think, I couldn't copy paste the full thing, but this is what we just spoke. And I think that was the last slide. Thank you, but I'll wait for questions. Dr. Sridhar, stop sharing. Okay.
Thank you, Avinash sir. So we'll be waiting for a couple of questions if any questions are there in YouTube. Else, I think I'll take the questions and send it out to Sridhar sir so that we can get the answers later on. Do we have any other questions for the YouTube channel? You can put it.
Okay, sir. We will check, sir.
Yeah, let me check. Okay, so I think we don't have much questions from YouTube channel, sir. So I'm very thankful for you for joining and being in a busy schedule. You have joined us and gave information which is very much easily digestible, and without any adverse event, we have got that information as cells, as ICSR, and the bigger versions as nucleus. And thank you so much for joining, sir. If any questions are there, I'll send it out to Sridhar sir for more information. Thank you, sir.
Bye.
Thank you so much. Thank you, everyone who have joined on YouTube. We'll be having one more last session by tomorrow, 5:30. Thank you, everyone. Thank you.